point and frameshift mutations. Okay, so let's start off by looking at some different types of frameshift mutations. Okay, so what a frameshift mutation involves the addition or deletion of nucleotides. Okay, so remember that a strand of DNA is made up of complementary base pairs. So if we look at a single strand of DNA, like this one here, you can see it's made up of G, C, now this is RNA, so it's got U, G, C, U, and A. Now, a frameshift mutation is the insertion or deletion of nucleotides. So for example, if we look at this one, we can see that on codon 5, if we look at specifically codon 5, so that's this one here, we have C, G, G. And then if we look here, the C and the G bases have been removed from the mRNA strand. So that's an example of deletion where the nucleotides are removed. Okay, so because those two nucleotides have been removed, we actually have a different sequence of DNA. So everything on this side has been altered and it's been shifted into the space where that C and G nucleotide were. So everything's kind of been moved or shifted like a frameshift mutation, and now the whole sequence after that error, after that deletion, has been affected. So that's an example of a frameshift mutation. Alright, because if we look at codon 5, now we have G, A, G instead of C, G, G. And that's affected, but also codon 6 will be affected as well. You can see now it starts with C, but before it was over here, so it'll have a different sequence of bases, and therefore code for a different amino acid. So that's an example of a deletion frameshift mutation. If we have a look here, so this one is an example of an insertion frameshift mutation. So this is our normal sequence up here, and the thing that's been changed is we have an extra T base that's been inserted into this first codon here. So instead of having ATG, we now have ATT, and this G has been moved over here. We can see it's here now. So we have GCA instead of CAT, and everything's been shifted one place to the right. So that's an example of frameshift mutation. Insertion, frameshift mutation. Now let's have a look at point mutations. So if an incorrect base is inserted and not corrected, that will result in a point mutation. So that's where a single nucleotide is changed, substituted, inserted, or deleted. So we looked at insertion and deletion and how that can cause a frameshift mutation here. But it can also cause silent mutation, missense mutation, and nonsense mutations. Alright, so if it's a silent mutation, so we have, instead of having if this is the original DNA that has been mutated, you can see in the silent mutation, the only base that has been substituted is the C. So instead of having UUA, we have CUA. Now that C, although it's a mutation, it still codes for the same amino acid. So it says up here that the original DNA codes for leucine. Now, the codon CUA also codes for leucine. So although there's a mutation, it's a silent mutation, which means you don't really notice it because it codes for the same thing. Now, a missense mutation or missense mutation, that's an example where the base is substituted and it codes for a different amino acid. So this one, GUA, codes for valine, so it'll have usually a greater effect on the organism. And if we have a look at the final one, the nonsense mutation, so that is when the mutation codes for a stop codon. So it's a specific mutation that codes for a stop codon sequence of amino acid bases. Okay, hopefully that explains point and frameshift mutations a bit more.